Hello, everybody. Today is going to be a class on cellular respiration. This is one of the most difficult topics in AP biology, and I decided to make this video to help the students to focus more on what we have in it. So the first thing we're going to talk about is the overview of cellular respiration. So we're going to talk about outside the mitochondria, which is where the glycolysis usually take place, because it does not need oxygen for its process. It's an anaerobic process. So the third one is fermentation process. Then we'll talk about inside the mitochondria. Then we'll talk about the metabolism. So cellular respiration is a cellular process that helps to break nutrient molecules produced by photosynthesis. Photosynthesizers are organisms that usually photosynthesize. So they carry out the process of photosynthesis with the concomitant production of ATP. ATP is like the coin of energy, or it's like the what you pay to get energy. So it is the kind of the currency which you pay where you need to get energy. So the meaning is the major reason why the process of cellular respiration usually takes place is to produce ATP. So it consumes oxygen and produces carbon dioxide. The meaning is the process of for the respiration would take oxygen because you got glucose plus oxygen, and it's going to produce CO2 plus H2O. So this is why it says it takes in oxygen, and it produces carbon dioxide as a product. So cellular respiration is aerobic process. It's an aerobic process. The meaning is it needs oxygen for the process to function. It usually involves a complete breakdown of glucose to CO2 and H2O. So cellular respiration involves the complete breakdown of glucose to CO2 and H2O, and this is what I wrote here. Energy is extracted from glucose molecule, and the energy which has been extracted from the glucose molecule is not something that happens one time. It's in stages. It happens in stages. So it happens in almost 10 steps before you can get energy being taken out. So it allows ATP to be produced efficiently. So ATP is being produced efficiently through this process of respiration which is taking place. So the energy is being extracted from glucose molecule in a stepwise manner. So it allows the ATP to be produced efficiently in the process. We have several processes of oxidation reduction taking place. So due to this reason, we need NAD plus and FAD. These are coenzymes that would help to facilitate the processes of oxidation and reduction. So this is a brief summary of the processes that happens when there is cellular respiration. There is oxygen, which usually come from the atmosphere, and there is glucose from the food these animals have taken into the body. So oxygen plus the glucose enters the cell, and they release H2O and CO2. So this is the mitochondria. The mitochondria is a organelle that has two membranes. So due to these, we have what we call the intermembrane. This is an inner membrane, and the other one is outer membrane. So in between the two membranes, we have a interspace. So we have what we call the intermembrane space. So this is the space here. So this is the space where we have an accumulation of ions, hydrogen ions, and some other molecules at the end of the electron transport chain reaction. So which we're going to talk about later. So the production of ATP is always happening in the mitochondria because it is the place, it is a location where the process of ATP production usually take place. So it uses energy from the glucose, which is being broken down to form ATP from ADP plus the phosphate. So every time energy is being needed, it does not take anything to make a ATP once you still have enough glucose to be broken down. It is only when you don't have any glucose anymore in the body that then it goes to produce ATP from all the processes. But we were going to talk about that in more detail later on. So the breakdown of glucose, which is a process that leads to the production of energy, happens like this. We've got C6H12O6. This is glucose. So we use oxygen to break it down. So in this case, when we break, oxidation is a loss of is a gain of oxygen, and reduction is a loss of oxygen. So in this process, hydrogen is being given off and there is a gain of oxygen, and that's why it's oxidation. And reduction here, hydrogen is being gained. So this is why it's reduction. So the meaning is glucose is being oxidized to form this, and 6-oxygen is being reduced to form this, and energy is being released. Electrons are removed from substrates. So, and also we can define it like this, the electrons, because we have what we call rig. So oxidation is a loss of electrons, because when you convert the glucose to 6CO2, there would be a loss of electrons. And when you where you convert oxygen to hydrogen, so there will be gain of hydrogen ion, which is a gain of electron. So this is why it says electrons are removed from substrates and received by oxygen, which combines with the H hydrogen ion to form water. Then glucose has been oxidized and O2 is being gained. So this is why we say the process of glucose to 6CO2 is oxidation, and the process of 6O2 to 6H2O is reduction, because hydrogen ion has been added to the oxygen molecule. Now let's talk about these coenzymes, which are very beneficial for the processes of oxidation and reduction. Nicotinamide adenine dinucleotide is a coenzyme of oxidation reduction. So it's an enzyme that easily facilitates the process of oxidation and reduction. It is oxidized when it gives off the electron. So, and it's reduced when it accepts electrons. So like I mentioned to you already, always remember this. Oxidation is a loss of electrons. So it's oxidized when it gives up, when it loses it. And it's reduction when it gains, when it accepts electron. And when NAD, when he does get, when he accepts, when he accepts electron, is going to be what is going to be reduced. So, and when it gains, when it gives off electron, it gives it out, it's been oxidized. So FAD is called flavin adenine dinucleotide. So it's also a coenzyme for oxidation reduction process. And sometimes NAD plus is being used instead of FAD. So in this case, it accepts 2 electrons. So NAD plus would accept 1 electron. It's going to accept 1 electron and 2 hydrogen ion to form NADH. So NAD, so NAD has been reduced. I want you to understand this concept right away. NAD plus is being reduced by gaining electron and by gaining 2 electrons and accepting, sorry, I made a mistake here. By gaining 2 electrons and gaining also 1 hydrogen ion. So it gains 2 electrons and 1 hydrogen ion. So this is what forms NADH. But as for FAD, FAD would gain electron. It's going to gain 1 electron and it's going to gain 2 hydrogen ion. So this is what is FADH2. So this is the way they form. So, but in most cases, this NAD is preferably used than FAD because this could be reused again. So every time you use this in the system, it can be reused over and over again. So this is why you have to make more of these in the system than this. And this is preferably used in a process than FAD. So now let's talk about the phases that is involved in cellular respiration. We've got to be very focused, and you've got to follow me up. So because some of these processes are very confusing, so you need to be very specific about what you want to say. Cellular respiration includes 4 phases. In some textbooks, you have 3 phases. So most textbooks will not put the preparatory stage. So they have 3 stages. So either you have questions in the quiz or exams that is you've got 3 steps that you should know this is being removed from it. But if you have 4 steps, you should know this is the second step. So cellular respiration includes 4 phases. Glycolysis is the breaking down of glucose to form 2-pyruvate. So what does that mean? Glucose has got 6 carbon atoms. So it's been splitted by using ATP as a source of energy. So it gives you 3C. So these 2 3C, we call them pyruvate. So you have 2 pyruvates from one glucose molecule. We're going to talk about that in more detail. So it occurs in the cytoplasm. Why does it take place in the cytoplasm? Because it's an anaerobic process that does not need oxygen for the process to happen. So in that process of doing this, ATP is also formed. So ATP is being produced, energy is being produced, because mind you, you had energy ATP being used, but it produces ATP as well at the end of the process. So the second stage is called the preparatory or prep reaction. What happens in the prep reaction is a prerequisite or is a preamble for the Krebs cycle stage, which is the 3rd stage of the reaction. So both the molecules of pyruvate, they are being oxidized to enter the matrix of mitochondria. Before, let me mention to you the places where they both take place. The glycolysis takes place in the cytoplasm. The preparatory stage takes place in the mitochondria, to be specific, in the matrix. So the Krebs cycle also, which we're going to mention after this, takes place in the matrix. And the 4th stage, which is the energy electron transport chain reaction, usually takes place in the Krebs state of the mitochondria. So both the molecules of pyruvate, they are oxidized and they made to enter the matrix of the mitochondria. So what happens is electron energy is stored in NADH. So all the electrons that have been produced when the breaking down of glucose is happening, so they've been stored in NADH to make it have enough energy at the end of the reaction, which needs to be used. So we've got 2 carbons which have been released from each of the pyruvate. I would explain to you the process later on, because when you have 3 3C, so it needs to be changed to acetyl-CoA. So acetyl-CoA it's got only 2 carbon. So what happens to the 1 carbon here is used to produce carbon dioxide. So the carbon dioxide, which is produced from each of these pyruvate, produces 2 CO2 and is being taken out of the body of the cell through the process of the fusion. So, but this is what has been mentioned here. We would get to a place where it's going to be discussed in more detail. So I'm just mentioning it to let you know or remember when we get to that place. So the third stage, which could be a second stage in some textbooks or some exams, is a citric acid cycle, which is another name for Krebs cycle. So what happens in this cycle, it happens also in the matrix, as I mentioned to you before, in the mitochondria, and produces NADH and FADH2. So this is a place where NADH and FADH2 is being produced because they need to be used in the last stage. So a series of reaction would release 4 carbon as CO2. So there will be a series of reactions that will produce 4 carbons as CO2. And that is a process whereby carbon has been taken off from the pyruvate. So this process will happen and continues to release CO2, which is going to release 4 carbon atoms. We're going to talk about that later. So it makes that times twice for a glucose molecule. So the meaning is 2 times for 1 glucose molecule to give you 4 each pyruvate. So if you have 1 glucose molecule would release, what am I saying, 6C. If you turn this to, it's going to become 3C, which is 3 pyruvate. So this one releases 1 CO2, this one releases 1 CO2. So this would happen again. So when this happens again, so this releases another CO2, and this releases another CO2, 1, 2, 3, 4. So this is why it says times twice for glucose. So it happens 2 times for every glucose. So to give you 4. So, and also this process, which we see here, produces 2 immediate ATP molecules per glucose molecule. So the meaning is for this glucose molecule, you have 2 ATP. So you have ATP, which is which is needed for the process. So when the production of ATP takes place, that means you have a total of 4 ATPs being produced. Mind you, you used 2 ATP before. So this is why we have a net of 2 ATP at the end of the citric cycle. So don't get confused. We're going to talk about that later in more detail. So now the electron transport chain which is called the ETC. It is a final process. So in this case also, this happens in the Krebs state of the mitochondria. So it helps to extract energy. Mind you, you saw the production of NADH and FADH2, and we told you before that electron energy are being stored in it. So now it's going to extract all the energy from NADH and FADH2. So it's going to pass the electrons from a higher to lower energy state. So there will be a movement of this energy from a higher energy level to lower energy level, because the energy will pass through the certain proteins until they get to a point where they need to be used. So this will produce either 32 or 34 molecules of ATP by a process called chemiosmosis. So we would see that in more detail in the next comment slide. So now we want to talk about the 4 phases of glucose breakdown. We want to use this picture here. Now, the first process, mind you, this is plant cell, this is animal cell, and this is plant cell. So, and now they show you that these procedures now in this mitochondria in it. So this is what happens in it in the process of glucose breakdown. So the first thing is glucose has been broken down to pyruvate. So in the process of doing this, it produces what? 4 ATP, as I mentioned to you in the process. So, and you have a 2 ATP net because 2 was being used in the beginning. So now the second reaction, which is a preparatory reaction, electrons are being produced in this reaction. And these electrons are being taken into the electron transport chain that will be used later on for chemiosmosis. Mind you, this reaction produces NADH, as this would also produce NADH. And also it produces NADH and FADH2, which need to be used in the electron transport chain reaction. So the third stage, which is a citric acid cycle, produces more NADH and FADH2, more electrons have been also produced. And the last stage, which is electron transport chain and chemismosis, so would not lead to production of 2 ATP or 32 and 34 ATPs. I will talk about what this means later on, because what happens is if there is no oxygen in the mitochondria anymore, what would happen is there will be fermentation. So fermentation process will start. So this will produce lactic acid plus CO2, or some alcohol will be produced. And if there is oxygen and the process of cellular respiration continues which is aerobic, so it produces 32 or 34 ATP. So this is what is being meant here. So we will see the processes in more detail later on. So now let's talk about outside the mitochondria, which is glycolysis. So glycolysis happens in cytoplasm, which is outside the mitochondria. So it consists of 10 reactions, each with its own enzyme. Mind you, we already know that enzymes are very specific in the reaction. So it happens in 10 reactions, and each of these 10 reactions have their own specific enzyme that will be utilized for the process to proceed. So now we want to divide the stages of this glycolysis into two. So how do we do this? So the first thing is we have 2 ATP. So we have one we call energy investment step, and we also have what we call the energy harvesting step. So I will talk about them one after the other, and we're going to have another future slide, which is going to be more detailed about it. So we have 2 ATPs in the beginning, as I mentioned to you, that are being used to activate glucose. Like I mentioned to you in the previous classes, every metabolic reaction would not happen simultaneously, would not happen by itself. And the reason why this would not happen by itself is to control the amount of it you have in the system. So energy is needed to activate the breaking down of glucose. So in this case, 2 ATPs are being used. So now, when they split up, they form 3GP molecule. So they form G3P molecule. G3P molecule. Why do we say they form G3P molecules? Because glucose is, let's say, is G6. Glucose has got 6 carbon atoms, isn't it? So when you utilize 2 ATPs, the phosphate molecule will be broken down and will be attached, because this G6 will be broken down into what? G3, G3. So mind you, you use 2 ATPs. So one phosphate will attach to one pyruvate, and the second phosphate will attach to the second pyruvate. So that gives you G3P molecules. So this is the process called energy investment. So energy is being used, that is to say, 2 ATPs are being utilized in this procedure. So now let's move on to the energy harvesting steps. So in the energy harvesting steps, so the G3P, there is an oxidation of it. So what does that oxidation mean? It means that electron is going to be removed and hydrogen ions also will be removed. So there will be a removal of hydrogen ion and there will be a removal of electrons. So what happens in that process? So that 2 electrons and 1 hydrogen ion are being accepted by this NAD plus, which we know is a coenzyme, and also to produce NADH. So now, as I've mentioned to you before, there will be several enzymes, electrons being released. So these electrons being released during the oxidation of these would be taken up by NAD plus to produce what? To produce NADH. So I told you before, so NAD plus would take 2 electrons and would take 1 hydrogen. We'll take 2 electrons and 1 hydrogen. So it's going to give you NADH, which is a coenzyme that will be used in the later reaction. So in that process, 4 ATPs are produced by substrate level ATP synthesis. So there is a section called the substrate level. So the meaning is the production of ATP to be used in the future. So an enzyme passes a high energy phosphate to ADP. So all the energy, high energy phosphate is being passed by an enzyme to ADP to make 4 ATPs. So what happens is there is not a net gain of 2 ATPs. Why? Because we actually use 2 in this process. 4 is being made in the second process, which is energy harvesting. So that gives you a net of 2 ATP. Mind you, both G3Ps will now be converted to what? To pyruvate. So the meaning is when they were G3P, we don't call them pyruvate. We call them a specific name, which we're going to talk about later. So it's an intermediate process to the production of pyruvate. So now they're being converted to pyruvate, which is going to have only 3 carbon molecules without any additional thing. So the input and output of glycolysis has been put here. So we've got 6 carbon of glucose, which is converted to 2,3 carbon pyruvate. So we had 2 NAD plus, which produces 2 NADH. So we've got 2 ATP, which has been used because it's been used, the phosphate has been taken away. So we have 2 ADP as an output. So now we've got 4 ADPs. We've got additional 2 more ADPs with this, plus 4 phosphate being carried by high energy enzymes. So to form 4 ATPs. So this is the way how you have 2 net ATP. So now the next one is showing you what happens, the procedures that happen in the production of ATP. So as I mentioned to you, this is BPG. It's got phosphate because of the enzyme with the ATP, which you use in the breaking down of glucose. So what happens is there is an energy, high energy phosphate, which is going to be added to the ADP to produce ATP. So this is how the production of ATP usually takes place. So that is what we call substrate level ATP synthesis. So now we want to talk about the energy investment step in more detail. So this is glucose in the beginning of the reaction. So the glucose would take 2 ATPs. So, and it's going to split the glucose molecules into two pyruvates. Each of the pyruvates would have as you know, when ATP is being used, it's the phosphate, which is the high energy phosphate that actually energize the procedure or the process. So this is why you've got phosphate molecule being attached to this. You've got another phosphate molecule being attached to that. So then in that process, 2 ATP is being used in the body. So what happens is now this is going to be splitted into what? Into 3 carbon molecules, which is G3P. So G3P, which is called the G3P is called glyceraldehyde 3-phosphate. Why is it called this name? It's called this name because it's called this name because it's actually a subordinate, is an intermediate process to the production of phosphate. So G3P is glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. So then we move on to the next step. So in the next step, what happens is there is a conversion, there is a production of NADH from NAD plus because we've released electrons from this process, and also we've also released hydrogen ion in this process. So the hydrogen ion and the electrons will be used to produce NADH. Now, 2 NADH have been produced, each from the pyruvate. So now there will be high energy phosphate. So there will be oxidation of G3P. So as these receive high energy electrons, so the oxidation would happen. Now, phosphate molecules are being added to the G3P. So that becomes what becomes BPG. So the BPG is a name given to this molecule because it's called one. This is carbon atom 1, carbon atom 2, 3. This is 1, 2, 3. So it's called 1,3 bis phosphate Bisphosphoglycerate, bisphosphate glycerate. So because you've got the phosphate molecules on atom 1 and 3. So as you see here, this is the high energy phosphate, which is actually been taken from the during the production, during the production of ATP. So which is going to lead to the production of ATP very soon. So this is why it's having this this wobble line compared to the other one. So now after this process of adding phosphate molecule to the process, so now we see boom, and ATP has been produced, and now it's gone. The phosphate molecule has been attached, it's gone. And now the phosphate here is now moved to where it was before. It was there before, but this higher energy will push it to the push it to the 3. So it pushes it to carbon number 3. But now that is gone, then it's going to take his place back. So when you're counting from the edges. You're counting 1, 2, 3, 1, 2, 3. So now, as I was saying, so now we've got 3PG. So the 3PG is called what is called a phosphate. It's called what is called a 3-phosphoglycerate. 3-phosphoglycerate. 3-phosphoglycerate is 3PG, because I want you to know that the bond was broken in the middle here. So if the bond is broken in the middle here, it's either you count 1, 2, 3, or you count 1, 2, 3. 1, 2, 3, or 1, 2, 3. So, but I think the way it's being counted is being counted from here, 1, 2, 3, and that's why you have, and this is also 1, 2, 3. And that's why you have 3-phosphoglycerate. So 3PG is 3-phosphoglycerate. You could see the high energy phosphate is being taken off to produce ATP, and what is remaining is a phosphate which was remaining from the ATP being utilized before. So the energy is still sticking to it. So here the energy, the phosphate you see here was being added to produce ATP, but this was being used, and that is what is being used to energize the process or the reaction to now. So now the next stage, you will now have an oxidation process that removes a water molecule. So the oxidation process will remove water molecule from 3PG. So what would happen now is you see the water molecule being removed, and you see another phosphate molecule is being energized. It's been made to energize the phosphate you had in the atom number 3. So what happens is once it's been energized, is ready to produce another ATP. So once it's been used to produce a second ATP, then we have the pyruvate without any phosphate molecule connecting to it. So this is why we have, within this process of energy harvesting, we've got 4 ATPs being produced, minus the 2 ATP you have here. That gives us 2 net gain of ATP in all the processes of glycolysis to produce 2 pyruvate. So this is 2 molecules of pyruvate, and the product is end product of glycolysis. Okay, now let's move on to the next stage.